Good morning. Welcome to the second annual Biotech Communications Competition. I couldn't be more thrilled to be here. My name is Dave Getz, and I am the owner and founder of CZ Strategy. We are a strategic marketing agency, and my gosh, I think we've worked with more early-stage startup companies than almost anything else we've done over the last 20 years. Biggest problem that you have with early stage is what? Landing new business. That first client, the first 10, the first 50, getting legitimacy in your space. So that's the business we're in. We work on the marketing side, creating lead generation for clients, and we're really excited to be a sponsor. I just want to say a big shout out to BioForward is such a, it creates this center of gravity for not only just our corridor here in Wisconsin, but really for the world. In a sense, everything's starting to move towards this area, but also just the minds and the science, and it's just a great privilege for me to be a sponsor. So thank you very much.
Thanks, Dave, and thanks so much to CZ Strategy for sponsoring this session. Hi, everybody. I'm Mike Harrison. I am the Director of Milwaukee Operations and Education Initiatives at BioForward. So this is the second year that we're doing this competition. It's a really great time to be able to organize this. At BioForward, we're always trying to find ways to engage students more with our industry. And a year ago, coming out of the pandemic, we had this feeling that, kind of this sickening feeling that the trust in the scientific community was really dipping. And how can we engage students while also kind of building this strength, this skill that our future scientists are going to need in order to keep building that trust. Later on, the afternoon keynote, they'll be talking about issues of equity and diversity and how they have to deal with the mistrust of the communities when they're trying to find people for clinical trials or blood donations. It's a real issue out there. And I think that scientists, especially young researchers, need to be on the front lines of that communication, but with the community. They need to be out there sort of demystifying what it is that they're doing in the lab and letting people know that what they're doing is important and critical work. So I'm pleased to be able to hold this second annual BioHealth Communications Competition, where you'll get a chance to see some of these young researchers and hear about the work that they're doing in a way that they're able to communicate with the general public. That's the goal of these presentations is to kind of take their incredibly complicated research and distill it down to a place where I can understand it, where everybody can understand it. That's the goal. So without further ado, I'm just going to call up the first person, which is Joshua Che. He's from UW-Madison in the Department of Biomedical Engineering. Josh?
Hello, my name is Josh Che. I'm an MD PhD student here actually at UW-Madison, and I'm in the lab of Bill Murphy. And today I'm going to talk about fossil-inspired minerals to stabilize therapeutic mRNA complexes at room temperature. So we all know that mRNA vaccination for things like COVID-19 have been really successful. That's kind of what brings a lot of us here today. And so we can think of this as being something where we take an mRNA that's encoding a specific factor, like the spike protein for SARS-CoV-2. We can package that into a lipid nanoparticle so we can actually form an mRNA lipid complex. And this we can package into a vaccine that can actually be delivered in the clinic. But one of the problems is that mRNA products are actually unstable, and they require specialized freezers to be able to be stored for shipment and actually storage in the clinic. And the problem with this is that you need really specialized freezers as severe as minus 80 degrees Celsius to actually be able to store these and ship them. So you have to be shipping them on dry ice to maintain the stability. And this really increases the cost of these products and reduces the amount of access, because you can imagine that only resourced communities will actually have these specialized types of infrastructure. But if we look to different types of materials from nature, we can think about fossils, which actually can preserve intact DNA for thousands of years. And it's the mineral components of these fossils that, when an organic material is fossilizing, can actually help stabilize the DNA dry over time. So our lab has actually developed fossil-inspired minerals, where we can actually take some sort of substrate material, use calcium and phosphate to mineralize on top to form a mineral-coated material. And in my work, I actually created 40 different types of minerals to screen stabilization properties to store RNA dry in a way that would mimic a fossil. And the way that I did this was that I used these mRNA encoding for reporter mRNA, which could glow green when it's added to cells or light up blue when it's added to cells, and actually freeze dry or lyophilize these and then store them at room temperature, 25 degrees C, to see how long we can stabilize this mRNA complexes at room temperature. And my approach used a lot of different strategies to improve mRNA binding, promote the mRNA stability with things like sugars, and also the study about the importance of how the material properties of all these different minerals can actually impact the stability of these mRNA complexes at room temperature over longer periods of time, over weeks to months off the shelf. And so what we found is that if we actually optimize these minerals, we can improve the storage and delivery of the mRNA using these minerals. And what we can see is that if we compare to the state-of-the-art technologies, we can actually store these mRNA complexes dry on minerals for up to 6 months at a stability of 25% relative to fresh complexes that are not frozen at all. And this is about 2.5 times better than anything else that is published in the literature at this time. And so if we're actually able to use this technology to improve the storage of dry mRNA, you can imagine that we'd be able to ship these mRNA at ambient temperatures without requiring dry ice across the world into under-resourced communities to actually improve the access of this technology and also reduce the cost overall. Thank you.
Thank you, Josh. The next speaker is Matthew Wilgate from UW-Madison in the Animal and Dairy Sciences Department.
Hello, everyone. I'm Matt Wilgate. I'm a second-year PhD student in the Department of Animal and Dairy Sciences. My research focuses on studying microbial infections, and today I'm going to be talking about the therapeutic potential of soil-dwelling amoeba against incurable bacterial biofilm infections. So biofilms are an accumulation of microorganisms embedded inside of a self-producing extracellular matrix. They allow bacteria cells to evade host immune recognition and become hundreds to thousands of times more tolerant to antibiotics. Approximately 80% of chronic and recurrent microbial infections in the human body are due to bacterial biofilms. One of the most common organisms that cause these biofilm infections are Staphylococcus bacteria. Staphylococcus bacterial biofilms are the leading cause of catheter-associated bloodstream infections and sepsis, demonstrating an urgent need to develop new therapeutic strategies against these infections. To investigate new therapeutic strategies against biofilm infections, we turned to soil-dwelling amoeba. Soil-dwelling amoeba are microorganisms, single-celled microorganisms that live in the soil, and they feed on bacteria as their primary food source. Although bacteria form biofilms in their environment to protect them from predators, amoeba have actually developed mechanisms to penetrate biofilms and consume the bacteria inside. UW-Madison is home to a unique collection of 1,800 strains of amoeba called dictyostelids. Professors at UW-Madison identified that multiple species of amoeba are capable of disrupting biofilms. And based on this work, me and another PhD candidate in my lab, Nathan Chesmore, were interested in seeing if amoeba produce compounds that are able to disrupt biofilm formation. So the figures here represent fully established Staphylococcus biofilms treated with compounds isolated from amoeba. As you can see in the figures, these compounds completely eradicated Staphylococcus biofilms. Additionally, we tested multiple species of these amoeba, and we found that multiple different species of amoeba harbor the same antibiofilm properties. Additionally, we found that These compounds are able to degrade biofilm in less than 2 hours. To assess the mechanisms of action that amoeba might be using to target these biofilms, we performed scanning electron microscopy. And from these images, these images suggest that amoeba are targeting the extracellular matrix of biofilms to release bacteria so that they can then feed on them. Additionally, we identified potential proteins that amoeba target in bacteria to disrupt biofilm formation. Biofilm-associated antimicrobial resistance and tolerance is a leading global health threat, and our research suggests that amoeba might provide new therapeutic strategies to target biofilm infections. Thank you.
Thank you. Next up is Mike Jindra, who is a student at UW-Madison in the Department of Biomedical Engineering.
Okay, hi everyone. My name is Mike Jindra. Thanks a lot for coming. And I'm representing the Flagler Lab from Chemical Engineering today. I'm going to start out with a question. If you had to find a needle in a haystack, how do you think you would do it? If you ask me, I would tell you just light the whole thing on fire. Now, after 4.5 years of engineering proteins, you will understand how I went from good, earnest first-year grad student, let's try everything, to burnt out, ready to graduate, let's light it up. So the enzyme that I want to engineer makes something that we call dodecanoic acid. Dodecanoic acid is something that's commonly found in shampoo, detergent. I'm sure you used it this week. However, this enzyme that I'm working with also makes some things that we don't really want. However, our goal is to engineer selectivity into this enzyme so that it only makes what we want, only makes the dodecanoic acid. And our vision is to one day supplant unsustainable sources of this chemical from things like palm kernel oil. So our question is, if we have an enzyme which does something good and it does something bad, how do we engineer it to lose that pesky side activity? Well, every enzyme engineer needs to know what we call the central dogma of biology. The central dogma is basically DNA makes RNA, and that RNA makes protein. And if we want to modify the building blocks of our protein or enzyme, or the amino acids, then we have to make the corresponding changes in the gene that encodes for the DNA of that enzyme. That's not too hard these days. However, there are 20 amino acids to choose from, and it just so happens that my enzyme is 350 amino acids long. So if I wanted to test every single variation, that's more variations than there are stars in the Milky Way galaxy. Not the best way to do it. First-year grad students think they can do it. They can't do it. So this is like going to our haystack and just looking for that needle by hand. But our question evolved to, can we use what we already know about enzymes to make this easier on us? So what we did is we looked at what other enzyme engineers did, and we said, can we do that too? It's similar to going back to our haystack and saying, other people found the needle here. We're going to look in that general location as well. So consulted literature, made changes that other smart engineers said worked for them. However, I still had nothing. I still had an enzyme that had this pesky side activity and no improvement in selectivity. But finally, I realized I could use the gift of fire. And that gift of fire in this metaphor is what we call directed evolution. Now, directed evolution is able to complete this mutational process in just a few hours, which takes nature millions of years to complete. And when you combine directed evolution with a good screen, what you can do is you can easily filter out all of the variations that you made that didn't work. So we did directed evolution, we combined it with a high throughput screen, and we were able to develop an enzyme which is 90% selective for dodecanoic acid. So in summary, we show that, while we still have a lot to learn about the underlying mechanisms of enzyme selectivity, we can use directed evolution to create more sustainable products today. Thank you so much for listening.
Okay. Next up is Michelle Roberts, and she is from the Medical College of Wisconsin, Department of Medicine.
Hello. As he stated, I'm from medical college. I am in a clinician's lab, Dr. Mike Widlansky, and I'll be talking about treating vascular disease in diabetic patients. So we know that diabetes is a metabolic disease or cardiometabolic disease, and it's usually characterized with high blood glucose. But this can be really detrimental to the inner lining of our blood vessels called the endothelial cells, leading to endothelial cell dysfunction. So diabetes can also lead to cardiovascular disease, which starts as this microvascular disease, progressing into macrovascular disease, which we can start to see in retinopathies or problems with eyes, or nephropathies in the kidneys, neuropathies in the nervous system, or open sores or ulcers that won't heal. And these could all lead to more serious cardiovascular diseases like stroke, heart attack, or even death. And a lot of us know that there's a lot of poor treatment options. I'm sure everyone has a family member or someone they know that's been touched by this disease. So we'd like to target the vascular endothelial cells for treatment, more specifically the mitochondria, or the powerhouse of the cell. They do a lot more than that, but we all know that they produce a lot of energy for our cells. And in diabetics, there's this off balance of mitochondrial dynamics. And we can see this in our human microvessels, which we can study under microscopes, and we can do other molecular techniques with. So like I said, in diabetics, there's this offset of the mitochondrial dynamics, and we can see this fission protein, FIS1, is highly expressed in diabetics versus normal controls. And this leads to a decreased network extent, and you can see this by smaller, more punctate mitochondria and fission versus fusion, which has elongated or more of a network of mitochondria. And our lab has already shown that reducing this fission protein can improve the vascular function. Specifically, we can knock down the FIS1 mRNA, and we can test the vascular function, which we see an improvement in the vasodilation in diabetic patients, which is endothelial specific, and this does not have negative effects on normal vasculature. So we're working to achieve this healthy balance to treat mitochondrial and endothelial dysfunction in diabetics. And taking this research a little further, my current role is seeing if the FIS1 protein acts alone or if it's in tandem with others that dispose of sick mitochondria in a recycling process known as mitophagy. So all in all, our main goal is to develop novel treatments for endothelial cell mitochondrial dysfunction, which will reduce this vascular disease burden and other complications. Thank you.
Okay, thank you, Michelle. Next up is Nathan Novi. He's from the University of Wisconsin-Madison in the Department of Biochemistry.
Great. Thank you for the introduction, Mike, and thank you all for coming and listening to my talk. Okay, so by the end of my talk, I hope that I can teach you what this is, convince you that it's interesting, and give one example of how scientists might engineer this organism to serve as potential therapeutics. So over the last roughly 100 years, we've been really fortunate to have antibiotics, which can be life-saving medicines against bacterial infections. However, the era of antibiotics is likely coming to a close. This is because of antibiotic resistance, which is a process that has reduced the effectiveness of many of our currently available antibiotics. Antibiotic resistance occurs because of occasional mutations in bacteria, which allow the bugs to slightly overcome the effects of antibacterial drugs. Bacteria with these beneficial mutations survive and replace the bacteria that cannot resist the effects of antibiotics. This process repeats, slowly allowing bacterial populations to get better at surviving in the presence of antibiotics, and is an unavoidable cycle when antibiotic use is widespread. A promising alternative to antibiotics is bacteriophage therapy. Bacteriophages, or phages, are actually viruses, and the term bacteriophage in Greek essentially means bacteria eater. This means that unlike the viruses that cause COVID-19 or the common cold, phages can only infect bacterial cells and are harmless to human cells. In phage therapy, bacteriophages are administered to a patient to target and kill harmful bacteria in the body, producing the same effects as antibiotics. Phages are actually the most abundant organism on the planet, and they are wherever bacteria are, which is everywhere. This diversity of phages is one reason they are such a promising alternative to antibiotics, but this diversity also creates a challenge for researchers, because scientists need to be sure that they use the correct phages when these viruses are applied as therapies. So, what do the right phages look like? Phages used as therapies need to target harmful bacterial species and avoid the many species of bacteria that are important for your health, like the bacteria that live in your stomach and help you break down food. However, isolating natural phages that are both safe and effective as therapies is very challenging and time-consuming. So, in my research, I consider an alternative approach. What if instead of finding phages that are safe and effective, we could engineer these phages in the lab? So this is a 3D printed model of a bacteriophage developed by the members of the Raman lab. And as you can see, it looks a lot like a spider. Its core components are this head that stores DNA and these arms that allow the phage to grab onto bacteria and infect its targets. In my research, I am modifying the DNA stored in the heads of these phages to change the structure of these phage arms and engineer the phages to target specific bacteria. One big challenge, though, is that there are a lot of ways to engineer these phage arms. Actually, there's trillions of different ways I could engineer these phage arms. And I don't have enough time to test all of these options. So instead, I apply machine learning to help me predict which phage arm variations are the most probable candidates for being able to target harmful bacteria and avoid good bacteria. To test my method of engineering these phages, I have designed thousands of variations of this phage arm that our machine learning algorithms predict will be able to target different strains of bacteria specifically, and I'm currently testing these engineered phages in the lab. My hope is that this research will help advance the application of phages as therapies, and that one day this research could help save lives in situations where antibiotics no longer can. Thank you.
Okay, our next speaker is Nishant Verma. He is from University of Wisconsin-Madison in the Department of Biomedical Engineering.
Good morning. We're a little bit like computers. Computers have wires running from their motherboards to their microphones and their speakers so that they can listen and speak. Similarly, we have neurons connecting our brains to organs throughout our body. There are differences. Our neurons are alive. They can regrow. But there are some similarities too. Just like a wire, our neurons carry electrical signals in them, and we can edit those signals. We can hack them to change organ function. This is the premise of bioelectronic medicine. For example, the carotid sinus nerve is a small nerve in our neck that transmit information from blood pressure sensors in our vessels to the brain. The brain uses this information to control blood pressure throughout the body. In a clinically approved therapy, a stimulation electrode is implanted next to the carotid sinus nerve, and an electrical current is delivered through the electrode to edit the signals on the carotid sinus nerve. And the response can be miraculous. A patient with the device implanted can walk in, have it turned on, and their sustained high blood pressure can decrease to a normal, healthy level within minutes. Now, unfortunately, this miraculous response is only the case for the minority of patients. In the majority, the stimulation dose or electrical current that can be delivered is limited by off-target nerve activation. and there are many off-target nerves in the region of the neck. These off-target nerves activate before the target carotid sinus nerve and lead to painful side effects, for example, unintended muscle contractions. To move bioelectronic medicine forward, we need ways to stimulate nerves that are more specific, more precise. This has been the subject of my thesis. I'm developing the injectrode, a novel stimulation electrode that is injected around the target neural structure and conforms to it, allowing for more specific activation. I've been running computational models to optimize and to show the efficacy of the injectrode. Then, I developed a technique to directly measure on and off target neural activation in vivo, in a living animal model, so that we can show that this injectrode is effective even in live tissue. I'm now using this target engagement framework in my final thesis project, as well as the tools developed in these first two projects to develop a current bioelectronic medicine therapy and to improve it in terms of its target engagement. So the therapy is applicable not to the minority, but to the majority of patients. Thank you.
Okay, our next speaker is Tawfiq Ali, and he's from University of Wisconsin-Milwaukee in the Department of Chemistry and Biochemistry.
I guess I'm going to take it out. So, good afternoon, everyone. My name is Tawfiq Ali. I'm a fourth-year PhD student in Dr. Pang's lab. The topic of my research is that I work with reactive oxygenase species, activated products. Today we will figure out how they function and whether we can use them as a combination strategy. So what you see here is a nitrogen mustard drug. For those who don't know what nitrogen mustard is, it was first synthesized in 1922 to cure warts in soldiers during World War. By 1940s, some of the countries found out that this was very powerful alkylating agent, and some countries even thought about using it as a chemical weapon. Luckily, it never happened. Good thing that I'm here working on this drug even now, right? 80 years later. So the compound itself, it's a nitrogen mustard, and some of the FDA-approved drugs are already existed in the market for more than 60 years. I don't know why it swapped. The nitrogen mustard molecule that you see here, what it does, how it functions, it alkylates DNA and it holds these two strands together, and this is what inhibits the replication process. So if replication process is inhibited, cells can divide. The problem with these two already approved FDA drug is that they're not very selective. So it will kill cells, meaning any cells. There is no selectivity because the reason people are still using it because the replication rate in cancer is often at a higher level, right? So you see more death among cancer cells versus normal cells. So the problem that we had is basically high off targets, lower selectivity, higher dose. Our goal was very simple, right? Which is derived from the problem itself. We wanted to minimize off targets. We wanted to improve selectivity and lower the dose. How did we do that? We just made a few modifications that took 10 years, right? So we changed this methyl group. I'm not sure if we have pointer here, but you see methyl group that says the improved permeability, right? So changing this methyl group only made a huge difference. What it did, it made the compound more permeable into the cell, which overall reduced the dose amount, right? So if it's easily getting into the cell, that means you don't need that much of the drug to be used as a dose. What about the other improved selectivity, right? Boronic acid, what does that group do? So let's look at that, right? So the compound itself is known as prodrug for the same reason that we have this boronic acid group. If the compound doesn't come in contact with hydrogen peroxide, it's inactive. But the moment it comes in contact with hydrogen peroxide, it gets super powerful at alkylating, like a Superman of the compounds, all the anti-cancer drugs, right? So what's happening here is it's coming in contact with hydrogen peroxide, and then it's causing DNA alkylation. Now, we try to screen it with 60 different cell lines, and we hit the jackpot, meaning we hit the cell line that is not curable at all. So far, we have no drug to cure the cell line named TNBC, triple negative breast cancer. This cancer is unique in a way that it's missing all the receptor that most of the commercially available FDA drugs or non-approved drugs target, right? So if it's missing all these receptors, how can you target cancer? Our compound happened to be 10 times more effective than already approved chlorambucil and all the other remaining anti-cancer drugs. So this has happened by luck, but it's good for us, right? Now, we also tested it on animals, right? You can see vehicle still has a higher level of tumor growth, whereas if you look at the treated compound, it shows that there is no tumor, right? So compound is super effective. However, even if the compound is super good, we are still trying to learn as much as possible to know about this drug. So my work was basically testing this compound and learning how it's functioning inside the cells. Now, we are also trying to see how it's affecting the DNA repair machinery, because alkylating agents usually affect the DNA repair machinery, right? So we are trying to learn something about p53 protein and also trying to diversify it so that it can be used overall throughout other cancer cell line. So in summary, this drug itself is 16 to 10 times more effective than already approved drugs. It's more effective in TNBC, where none of the drugs are effective. Also, we minimize the dose to almost 10 times and improve the permeability, improve the selectivity, got rid of a lot of off-target effects, and this is a potential candidate for a combination therapy. So I am in a process of using it with another ROS enhancer that could be used selectively towards cancer, and then we can enhance the ROS level, meaning hydrogen peroxide levels, right? And then make this drug even more diverse. Now, the whole criteria of my project isn't just talking about my drug. There's a good cause towards this project, is that only 13% of the 15% breast cancer cells is the market for this drug. So our group is focusing on that minor section of the patient that nobody cares about. Thank you.
Okay, and our last speaker of the day is Veda Patil, and she is from the University of Wisconsin-Madison in the Department of Biomedical Engineering.
Hi, good morning, everyone. My name is Veda Patil, and I'm a PhD candidate in biomedical engineering in the master's lab. And I'm a PhD candidate in biomedical engineering in the master's lab. So we all know somebody who's been affected by heart disease. It's in fact the leading cause of death in the US. And my research is about a disease that you probably didn't realize you had to worry about, which is calcific aortic valve disease, or CAVD. Up until some 15, 20 years ago, we didn't realize that this was an active disease. We thought it was a side effect of aging. Now that we've established that it's a life-threatening and active disease process, we want to find some solutions. So CVD is a disease that affects the valve, which is housed within the aorta, which is the largest blood vessel in your body, and it directs the flow of blood. Now, if this valve becomes too stiff or unable to function, this can lead to heart failure. Unfortunately, this disease affects 25% of adults over the age of 65 who will exhibit some characteristics of this little-known disease. What's worse is that there are no treatments currently available. The only thing you can do is get a total valve replacement. So, to be able to design future treatments, we need to be able to map the pathways and the molecular interactions that govern CAVD progression, which is the motivating factor for my research. So I look at how risk factors like smoking, a bad diet leading to high cholesterol, diabetes, aging, and biological sex might contribute to CAVD progression and how it affects these pathways. Now, good news for me, but bad news for those of the male sex. Men have an increased probability of developing CAVD. And moreover, this disease presents differently in males and females. So as you can see, females tend to have more thickened valves with scar-like tissue, And males tend to have more calcified or bone-like deposits on their valves. And that's for the same level of disease progression. So what causes the difference in outcomes for males and females? And why is CAVD less prevalent in females? Well, we thought that the answer might lie in sex hormones, specifically estrogen in females. So to investigate, I took some cells from pig hearts and I treated them with estrogen and a stimulus that will cause CVD within these cells to see how these cells respond. And what we found is that estrogen presence can actually mitigate the buildup of proteins within the valve that cause it to become thick and stiffened. And we also identified some potential mechanisms that might be involved. So overall, being able to map these pathways will help us develop future treatments or prevention plans that will affect the millions of people that are affected by this disease. Thank you.
Okay, well, another round of applause for all of our speakers today. And special thanks to the last two presenters for dealing with some weird PowerPoint issues that I apologize for. So to kind of back up and give everybody a little bit of background on these presentations. So the students submitted a 3-minute video to BioForward, and this happened weeks ago, and several board members of BioForward judge these on a pretty rigorous judging sheet, and so really they came here just to present this to you, but the judges have already decided the winners. So we even though here at BioForward we're very idealistic and supportive, we know that we do have to, if we're going to get extremely busy students to take time out of their day and show up here and present to everybody, you got to give them something. So everybody that is presenting today receives a $150 gift certificate to Amazon, and the top 3 winners win cash prizes. The third is $500, second place is $1,000, and first place is $1,500. I'm going to read those the top 3 videos, and if those students will come up here and join me for another round of applause. So in third place, we have Mike Jindra. Right there. In second place, we have Nathan Novi. Stay right there. And in first place, we have Veda Patil. All right, well, another big round of applause for our winners. Okay, you can have your seat. I won't make you stand here while I talk. So that will wrap up our session here today. So you have a little time if you want to talk to some of the presenters. It was a great group this year. The judges were very impressed by all the videos. It was a very diverse crowd of topics that they're discussing. And that just shows you that it's just scratching the surface of the kind of work that's being done at Wisconsin's research universities. So after the session here, we have a lunch session, where we will be giving out our awards for scientific and business achievement. So if you would kind of make your way down to the lunchroom, it's a buffet style. Grab your food, sit where you'd like, and network with some of your fellow BioHealth community members. Okay, thank you very much.